So I want to first remind everybody that there is this nice YouTube video available. And this YouTube video is pretty short and sweet, all about the antibody-mediated immune response. Remember, we can also call this the humoral response. And it's really all about like activating B cells so that they can differentiate into plasma cells and memory cells. And the memory cells are going to be what allows us to clear pathogens sooner when we encounter those same pathogens in the future. And the plasma cells are the specific type of cells that produce antibodies. Now, before we move on to cellular immunity, I want to do a recap of the two diagrams that we've talked about in the past few videos, which is going to be the phagocytosis diagram and the antibody-mediated response diagrams. Let me put those on the screen. Sorry, it can't be full screen. I'm trying to outsmart PowerPoint. When you have a presentation going, they have really good subtitles here. So I have to put the PDF over it so you can actually still see the subtitles. Anyways, I digress. So we talked a lot about phagocytosis before and how the main goal is for the phagocyte to find, attach, ingest, and digest, and then clear the pathogen. Well, that is true of antigen-presenting cells. However, there's an additional 2 steps involved because an antigen-presenting cell is going to do what the name implies. It's going to present antigen, and it gets that antigen by digesting the pathogen, okay? But in order to present that antigen, it has to be able to display it on its surface. Well, the only way to display an antigen on its own surface is to attach that antigen to a molecule that naturally belongs on its surface. And so that's where MHC2 molecules come into play. Remember that all antigen-presenting cells have MHC2 molecules on their surface. And so, as you can see in this diagram, here in the middle, part of the phagocytosis process when presenting antigen is to make additional MHC2 markers, merge them with the residual body, because remember, the pathogen at this point has already been digested. And so then look how the MHC2 and antigen combine together, right? And so now when the rest of the pathogen debris is released from the cell, these antigen MHC2 complexes will also be released, but they will go to the edge or the surface of the cell where the MHC2 belongs. And so now this dendritic cell is going to be presenting that antigen on its surface because the antigen is attached to MHC2. So let's walk through this diagram again very quickly. So here we have our pathogen. We're just going to say it's a bacteria in this example. The antigen is specifically those pointy triangles on either end of the pathogen. So in this part of the diagram, the pathogen has already found and attached to the pathogen. It's in the process of ingesting it. So it ingests it, and so now it's in this little vesicle called the phagosome. And as you can see, the lysosome, which is going to have those hydrolytic enzymes, is on its way, and it is going to fuse with the vesicle containing the pathogen. And then now that the hydrolytic enzymes are able to access the pathogen, it will chop the pathogen up into lots of tiny pieces. So now you can clearly see those triangular antigens. So notice, so remember, we call this the residual body. It contains all of the digested pathogen. Well, at this point, some MHC2 markers have been created, and it's going to merge with that residual body, and that's what's going to allow the antigen and MHC molecules to bind together, And then that vesicle is going to be exocytosed. It's going to use exocytosis to get to throw everything out of the cell, in which case the MHC2 antigen complex will go to the surface of the dendritic cell. And that's important because we're looking across the top here, okay? We're looking in the middle specifically at the top. So that dendritic cell now has the antigen MHC2 marker. And the reason that's important is because T cells can only recognize digested antigens. And so a virgin T cell that has its own little receptor on it is going to be able to bind to that antigen MHC complex, and that is going to allow little cytokines, little molecules made by the dendritic cell, to allow those cytokines to activate the T cell. So this virgin T cell is going to then differentiate into different types of T cells. The one we're concerned about here is the helper T cell, because as the name implies, it needs to go and help activate our B cells. Because with the humoral response, B cells are what we really care about, okay? But it turns out we cannot activate a B cell unless we have the help of a T helper cell. So that's kind of what's happening along the top. The dendritic cell bound to the pathogen, it phagocytosed the pathogen, and then presented the antigen on the surface of its cell, which was recognized by a virgin T cell. This binding is going to activate the T cell to to differentiate into a T helper cell. Okay, so that's part one of the story. At the same time, we have virgin B cells that are circulating throughout the body on the hunt for their specific antigen. And when it recognizes the pathogen, notice it is recognizing the entire intact pathogen. That's a big difference between B cells and T cells. B cells recognize the entire intact, undigested pathogen, whereas the T cell could only recognize the digested pathogen or the digested antigen. Okay, so we have our virgin B cell. That is an antigen-presenting cell, so it has an MHC2 marker as well. So that intact pathogen is going to bind to the virgin B cell. That virgin B cell is going to phagocytose and present that antigen. Okay, so a very similar process like we saw with the dendritic cell. Okay, but now this B cell is activated because it has that antigen presented. Okay, so now we're looking inside the box in the top left. So we have our activated B cell that has the antigen presented. So now look who's going to interact with that B cell, that T helper cell, okay, that we activated in the top portion of this diagram. So that T helper cell is going to bind to that digested antigen that the B cell was presenting. This helper cell, this T helper cell, now that it's bound, is going to release cytokines that are then going to induce division of this activated B cell. Okay, and so as you can see, an activated B cell, once it has been induced to differentiate, it is going to differentiate into either memory B cells, into memory B cells and into plasma cells, okay? And so the plasma cells are going to be what releases all of the antibodies, whereas the memory B cell is going to play a role in our secondary immune response, okay? So again, just kind of a big overview, the dendritic cell is presenting antigen in order to activate the T cell, which will form T helper cells. And as their name suggests, they need to help. And so they help by binding to an activated B cell. Because when the T helper cell and the activated B cell interact, then the helper T cell releases cytokines that triggers cell division in the activated B cell. That's what's going to allow it to divide and differentiate into both memory cells and plasma cells. Okay, the plasma cells will make antibodies, the memory cells will make it so that we don't get sick every time we are exposed to this pathogen in the future. All right, y'all, I hope this video helps kind of break down this very involved, complex diagram. Please let me know if you have any additional questions.